forms of the diseases over here. So actually these are quite smaller than the actual market for heart failure or cancer or HIV. They're very disease specific. But we can see that the market's growing and we're seeing a lot of money go into this area. Imagine if you were making therapies that affected everyone on the planet. You think we make a lot of money? Well, we probably would, but our goal is actually to bring the price of these therapies down and give these therapies to everyone. To make them so inexpensive that no one is barred access. We believe that they'll be given like immunizations, that you'll get your gene-boosting therapies at specific times in your life to ensure that you don't get sick. Right now, we'll vastly have to work in the elder and the sick. It's disease states that governments think are acceptable for us to try these experimental therapies. But we'll work them back younger and younger, and we're going to blur the lines between preventative medicine and physical enhancement. These are some of the future consequences, some of the things that you can get excited for in the future. Maybe some things that will come down the pipeline, hopefully in your lifetime. Organ regeneration in the body. We believe that some of the gene therapies can do this. Radiation resilience. I've actually been contacted by NASA and other people, other companies who are looking to go to space, and they're looking for resilience to radiation for astronauts, which actually gets on space technology down here. Better vision, intelligence, physical enhancement, nutrition. Everyone has the gene for vitamin C. We could be creating our own antioxidants. We just need to turn it on. Autoimmune disorders and congenital disorders. One thing I'm really excited about is digital interface. The biggest computer companies in the world now are starting to code in DNA. What does that mean? One thing it means is that you can hold a lot more data on DNA than you can in the old binary systems. But it's quite possible that in the future, instead of holding your iPhone, it might be integrated into your biology. That would actually be pretty cool, and you wouldn't forget where you put it. So this is kind of a crazy look at the map. My slide is a little bit messed up. Right now, my company is traveling the world. We're building a coalition of partners. No matter what you've read, our team is getting bigger. More countries are behind us. More governments are talking to us at the highest levels. More scientists have connected with us. More companies have contacted us to try to get us to launch their therapeutics into humans. But we need everyone's help. And this is the sobering moment. Today, 100,000 people will die of aging. Not to mention the children that are dying of other diseases. That means 40 million people a year. That's a lot. And it's actually a very expensive lot of people. 60 to 80 million people this year will suffer of diseases with no treatment, absolutely no treatment. And because of the myths that we believe, things called regulations, the right way to do things, these people will die with no options, none. Even though we've reversed aging in mice, even though we can build muscle in human models, even though some of these gene therapies are through safety and efficacy, these people have no options because the gene therapy is only run for a specific disease. We're going around the world and asking countries to let down their barriers, to bring in new regulation, to stop with the old bioethics that's not very ethical, that is killing people, and bring in the new bioethics of allowing people to be pioneers. We want to take the names off of the map, create one civilization that's fighting the only enemy that we know, disease. I hope you'll join us, and I look forward to your questions. I believe that Mr. Abbey Roy will be talking next.
So thank you very much, Liz. And now I would like to
give the floor to Abby Roy, president of BGRF, Biogerontology Research Foundation. Хотелось бы сейчас предоставить слово Ави Рою, президенту фонда исследований биогеронтологии, создателя и редактора издания Longevity Reporter, и также член управления Международного альянса продления жизни.
Thank you, Daria. Right. It's always intimidating following Liz. I think we should switch the order around. I'm going to lower some of your standards now. But I think my presentation at least follows really well after Liz. One thing that both Liz and I would like to do is to have a conversation this evening, besides just Q&A, about how all the things that we are showing you, we do it better, cheaper, faster. And I think there are fantastically clever people here that can help us do that, so enable it together. Everybody here has heard of anti-aging clinics and private clinics and things of that nature. This is not going to be anything like it. This is a project that came out of thinking about what Liz was doing and how to accelerate that. So when Liz did her therapy, the main questions that arose is, how do you know that there's a difference? Since Liz didn't turn into a Disney fairy princess overnight after taking her therapy, how do you tell that there is something going on? More importantly, how do you capture each one of you here and understand what is going wrong with your tissues, your organs, and at the cellular level, so that we can actually use some of the therapeutics that we are developing in labs to treat your condition. So the precision and personalized medicine clinic, preventive medicine clinic idea came from that. If we can use the latest technologies to treat, to understand what is happening to you better, then we can design perfect treatments. I think the mainstream clinical trial organizations are also coming around to this same idea. The more precisely we can measure what's happening in your body, the better we can choose existing drugs, which don't clearly work in most people, and upcoming therapeutics to treat you. In order to do that, we do this, which is break you down into every single layer that is possible. So all the way down from your genome to all the way up to the environment that you interact with every day. So right down here, these are terms coined by Eric Topol. If you read his books, those are fantastic. The Patient Will See You Now is his latest one. So this is exposome, which is the interaction of your genome and the environment. This is the epigenome, microbiome, metabolome. So the idea of our clinic is to collect every single piece of this data. These will be more familiar: functional magnetic resonance imaging, CT scans, and neuronal mapping, and so forth. The idea here is nothing special. Each one of these individual layers, you can actually go and do it for yourself. But how do we convert that into clinical practice? That is the main goal that we're trying to solve, is putting all of this data together so that we can have a clinical output, so that we can understand how your body, yours, not someone else's, is aging, at which part do we need to treat, and which parts can be left alone. Where do we think things are going wrong? So that's the first picture that you would get, and that's simple diagnostics. Not really simple, but simple. The next part is putting all of that data together, putting it all together and predicting what might happen in the next 10 years, in the next 20 years, in the next 30 years to you. And doing that progressively. As the data comes in, we move that forward. We have 500 different markers that we are looking at for predicting your aging outcomes and your aging scores. So ultimately, we would be giving scores for each organ and various tissues and the combinatorial age that would look at your biological degradation over time. And that is the prognostic part. Finally, all of that comes together. Okay, this is not rendering properly, but what you would see here is the current failure rate in pink for the various drugs. I can give you a summary statistic of the drugs, cancer drugs available for treatment, 71 of them in the last approximately 12 years, 12 to 14 years. At $9,000 per treatment, they improve your life or increase your lifespan by 2 months. That is the kind of effectiveness we're talking about with current medicine. So what we would do with your personalized diagnostics and prognostics is then select the right treatment. And we're partnering with companies like Bioviva, and we are looking at other advanced cell therapies because the small molecule route isn't working. Along with pioneering cutting-edge diagnostics, which everybody is more than happy to do, and cutting-edge prognostics, we are going to, for the first time, pioneer cutting-edge therapeutics. That's the aim here. And again, we want to do what is best for the patient. So we're not actually going after any one particular hypothesis, besides just showing you how your body is breaking down. This would be the way that we would present your data. This is Microsoft's HoloLens, which is something that we're partnering with. You would have an augmented reality presentation of your imaging data coupled with your omics data, all of your metabolomics, your proteomics, your whole genome, and transcriptomics. All of the data that we would gather, you would be presented in this form. This would be the doctor working with you to deliver treatment to you. And this would be something that you would be able to understand, not just reading numbers on a chart. And this value is up or that value is up. So you would be in control of your own health. One thing that we were constantly asked is that my organs inside are getting younger, but do I look younger on the outside? That is the most important thing for many people. Obviously, we would be working with skin. Skin is your largest organ, and if we cannot treat the skin, we obviously cannot treat many other organs. But again, with skin, it would be personalized therapies. So something that we have currently that we're working with is personalized stem cell therapies for your skin. You currently probably use creams, but what if it could be made from your own stem cell factors or your own stem cells? That is something that we're working on. Again, it would be personalized and tailored to your particular needs. This is something that 23andMe currently does a little bit. If any of you here have a 23andMe profile, every time you log in, over 2 or 3 weeks, you get new information about your SNPs on 23andMe. So you get, every time medical data is updated, which is every 5 minutes, what we would do is systematically analyze your data and produce and give you more input into what is happening. So as science progresses, the more information you will have about yourself. Biobanking is a very, very important thing, and the clinic would involve biobanking with it. So every time you come to the clinic, which is at least 4 times a year, we would store your MSCs, your mesenchymal stem cells, your hematopoietic stem cells, and anything else that we can get a hold of. Because the sooner you store, the less damage you have on those tissues, and the better we can use them in the future. Also, we would be biobanking your gut microbiota, which is something that is unheard of currently. Nobody really does that, but that is a very, very important part of the puzzle, is the bacteria that live on you and in you. Emergency services is something that we absolutely want to pioneer. We're working with both equipment manufacturers and software companies to design things that can detect stroke before you have stroke. Everybody, or many people here, have those wristbands that tell you your heart rate monitors and your heart rate variability, but it could be great if you could predict if you were going to have an incident that could actually impact your life. And that's what we want to do with emergency medicine, is to predict if something is coming up over the next weeks, hopefully over the next days even. That will be an advancement. And if something does happen, this will give a call out to the appropriate authorities. This is something that would be very useful for the elderly patients, but certainly for everybody. We, like Bioviva, we're partnering with clinics all over the world. We are having discussions with our colleagues in Oslo, in Stockholm, in Helsinki, in Guernsey, in Austria, and in Russia, to bring these technologies, share the technologies amongst us, share the patient population, and deliver as quickly as possible the therapeutics to patients all over the world, bring down the costs, and deliver to as many people as possible. Currently, obviously, the costs are going to be high or high-ish, but it's going to provide technology and benefits that nobody has ever seen and no clinic is offering in the world. This is just a small part of our team, of our startup team that is based mostly in London. And these are some of our partners. What I want to reiterate is that we don't have a hypothesis. Many times when you go to an anti-aging clinic, no matter where you are in the world, they have a particular treatment to sell. We have no treatment, no one particular treatment to sell. We will first figure out what is it that is happening, which part of the aging process is damaging which part of your tissue or organs, and then we will work towards treating that. And we have partnerships all over London in order to facilitate that. And we're building partnerships in Oslo and in Stockholm and in Helsinki and Mumbai and in Dubai, so that we can find all of those partners who can help us treat our patients better. And Liz and Bioviva are certainly one of those partners. So that is the Precision Medicine Clinic. And before I wrap up, I want to quickly mention that on this poster you see Global Health Span Policy Institute, and Daria mentioned about the International Longevity Alliance. These are the organizations that we're working with so that we can bring the most cutting-edge technology in the most responsible manner, in the most regulatory or embedded in the regulatory framework manner that we possibly can. Of course, certainly Bioviva is working in that same environment. But we are trying to forge ahead with the Global Health Span Policy Institute in Washington, in London, and everywhere else in the world, trying to discuss with governments proactively about bringing treatments to patients. Yet another thing that I think clinics all over the world should be doing, because that is the interface between the lab and the patient. And that is where the Global Health Span Policy Institute and International Longevity Alliance come into the picture. Thank you for listening, and of course Liz and I will be here for Q&A sessions.
Спасибо. Thank you so much, Evan. Thank you so much, Liz. Дорогие друзья, давайте, может быть, мы так сделаем. У нас будет, например, 10 или сколько нужно уточняющих вопросов, а потом перейдем к дискуссии, к высказываниям. Кто-то, может, про свои расскажет, впечатления, проекты и так далее. Давайте тогда еще, Михаил, нужно тогда еще как вторым микрофончиком нам? Ну, вопросы, квиз, ковид, Кариксу, кому угодно. Вот тогда. В порядке или мой, может быть, дать микрофон? Давайте пока нажимаем микрофон, когда это включающий. Ага. Включаем, там что-то есть.
Добрый день, меня зовут Александр. Вопрос к Лиз.
встрече. Возьми стульчик, может быть. I'll just pass the microphone as needed.
Добрый день, меня зовут Александр. Вопрос Лиз. Вы в своем выступлении говорили о лечении достаточно большого спектра заболеваний геномным методом. Хотел спросить, есть ли у вас какие-то исследования с заболеваниями, так называемыми аутоиммунными заболеваниями, вот этой областью? Можно про это подробнее? Спасибо.
I, of course, have a huge interest in autoimmune diseases. That's what diabetes type 1 is. Many of the autoimmune diseases are multigenic diseases, meaning there's more than one gene that's affected that's tipped off the system. We're hoping, as far as the immune system, that telomerase would go far to creating a youthful system that will help in some ways. But as far as multigenic mutations in the genome that cause things like diabetes type 1 and Crohn's, we're still greatly trying to find out through the research which genes are responsible and how to deliver them. In the future, we'll probably use CRISPR technology to do that. Abby, do you have any other responses?
Yeah, I think, I don't know if this is, yeah, okay, this is, yeah. So autoimmune diseases. Yeah, okay, so for cancer therapies at least, immune system modification is being tested as a treatment, as a method of treatment. And as Liz said, I think gene editing probably would help much more than turning on or off genes in the autoimmune system. So that'll be definitely useful for the future, but not currently applicable technology immediately available.
Возможно, у вас есть какой-то прогноз по срокам развития данных технологий именно в этой области?
When you think of Bioviva, you need to think of it as a technology company. So what we're doing is bringing therapeutics with the greatest amount of research and promise to humankind to clinic. We still depend just as much as before on universities and researchers making new breakthroughs in innovation. As our company gains momentum, we will go into research and development ourselves. But for your very specific questions, looking at universities that are already driving forces in these diseases is a great place to start. And if you see a therapy that looks like it's developing out of animal model to human, give me a call and we'll see how it works in humans. And Abby brought up a good point. Immunotherapy is coming along really quickly, and even outside of genetics, as far as cancer goes, trying to hunt cancer through mutations in your genes may not be the best route. Not every disease will find its best route through simple gene therapies. Sometimes looking at the mechanism of the immune system itself and creating immunotherapies that rely on the reaction of the immune system itself and create boundaries between the body and certain tissues will be of best magnitude. So we'll just continue to watch these, and if they look promising, we'll bring them to the market.
Здравствуйте, меня зовут Евгений. Я хотел бы, во-первых, поддержать вас в том, что иммунная терапия – это, конечно, будущее. Терапия, прежде всего, РНК. На сегодняшний день 1991 одна субстанция зарегистрирована в FDA. чуть больше 200 субстанций низкомолекулярных, чуть больше 200 субстанций зарегистрировано как белки и пептиды, и, по-моему, только одна как РНК. Естественно, вектор совершенно очевидно и будущее за этими препаратами. Но я бы хотел у вас узнать, а как далеко вы продвинулись на стадии доклинических испытаний? Что показали испытания на животных? Пирогенность, мутагенность, тератогенность? Что можете сказать об острой токсичности, хронической токсичности? До какой стадии вы дошли? Кролики, в конце концов, так сказать, все должно закончиться приматами. Вот в какой стадии вы сейчас находитесь? Что вы доказательно можете? Спасибо.
Okay, I'm gonna talk slowly so that translators can get this through to you. We are a technology company. The research has already been done. In telomerase, it has been used in a million studies. It's been used in mice. It's been used in human cells. It has over 10,000 peer-reviewed papers behind the therapeutic. The myostatin inhibitor, the muscle-building gene therapy, has been used in every animal model, including monkeys, and is in phase 3 in humans with muscular dystrophy. Safety and efficacy has been proven. Safety and efficacy is being proven in the other gene therapies. Our company now has raised money for one reason, to start clinical trials. We will now gather the data to find out what happens in humans, sick humans, moving back to healthier humans over the years in preventative medicine to create a healthier society. So we vastly have to remove the premise that every biotech company is a research company. As a matter of fact, if these gene therapies don't work, we'll work with other ones that do. We are going to continue to bring products to the world so that you have an outlet to get them. So we will not use anything that doesn't have peer-reviewed research. We will not use therapies that don't have mouse studies. What Elena's doing is massively important. Our company is not going to waste money and repeat those while people are dying today. There's no reason to do that. So you're welcome to get in touch with us, and we can send you the myriad of papers that are behind these therapies, and we would like people to look at them.
Еще вопросы?
Hi, I'm Nikolai Duchkov. I would like to ask Liz and Dati, and I have two questions. Okay? First of all, I would like to ask you what kind of changes in the regulatory systems would you like to offer? Okay?
Okay, Abby can probably talk more on that because he works more closely with that group.
Well, with the Global Health Span Policy Institute and with other consortium that we are working with, including groups at Oxford University, something that we work on daily is to see what we can do in order to bring therapies to market. I think if you go to Nature, the journal, the magazine, one of the latest articles points out precision medicine, a clinical trial of one. And ultimately, and that article, by the way, has a very nice picture of how the drugs, the top 10 drugs that are selling right now in the United States, how many patients they don't work in. Somewhere between 25 to 75% of the patients, those top 10 drugs don't work. They don't work well. Why am I saying this? We need to use the technologies that I showed you, precision and precise and personalized medicine, in order to reduce the clinical trial numbers so that you're delivering the right treatment to the right person so that you can see the effect difference. So to succinctly answer your question, what would we need from the regulators? Well, what we are discussing with the regulators in England and in other countries is let's use precision and personalized medicine. President Obama has a precision medicine initiative. So does our colleagues in England. And we are going to use that in clinical trials to reduce the number and the time so that we can see effectiveness as quickly as possible, and we can come down to possibly n=1 experiments. So we need regulators to use the latest technology to get as much benefit as quickly as possible and do human trials as quickly as possible. Additionally, I do not know who said this, but it has been cited that Albert Einstein said it, but I'm going to say it anyway. Doing the same thing over and over again, expecting different results, is a sign of madness. 99% of the clinical trials fail between phase 1 and phase 3. Okay?
Okay.
Do you know what? So the preclinical trials leading up to phase 1, we want to change that as well, as quickly as possible. So use the technologies that are currently available. So if we're going to ultimately test on patients like you, which we have like 10 of, let's say, some similar patients like you, why don't we do the preclinical trials on your cells that we can take out of your body and do it on them? So again, technologies. So we want to get into humans as quickly as possible, even if it is human cells growing on a plate on a 3D culture.
Thank you. My second question is completely connected with my first one, and I would like to ask you, what do you think about the problem of multiple comparisons? Okay? Because when we are working with such amount of data which you offer to work with, we have will have some issues with testing of statistical hypotheses. That's why, it's, of course, it's widely discussed in clinical and non-clinical trials. And what do you offer to do with that?
Okay, so I think there are two parts of your question, and this is getting very entertaining for me. So you're talking about, we have all this data. So let's just take transcriptomics, okay? So I pull up a transcriptomic data set, and there's 20,000 genes. Oh, sorry, all right, sorry. So I pull up a transcriptomic data set, and there are 20,000 genes, let's say. Now, if your null hypothesis is 5%, which is what you were getting at, you would randomly find 1,000 genes, randomly find X number of genes that are variable. Well, that's because you have a Fisherian world with you. I come from Oxford, I don't come from Cambridge, which is where Fisher did his work. Anyway, but my point is that you can get away from that if you are not looking for confidence like that. I think in biology, there's tons of articles about this, by the way, which is how you can look for 95% confidence interval and find that. We're using Bayesian methods, which is far better. So you have a hypothesis of the world, and you use yes or no at every step. By the way, DeepMind, all of those Google technologies, machine learning, they're also using Bayesian. Ultimately, the world is Bayesian. When you make a decision, you don't use a 95% confidence interval whether the chair was there or not. You use a yes or no. You have a significant confidence that the chair is going to be there when you stand up and sit down. So Bayesian hypothesis testing can eliminate all of that uncertainty from that data. We don't have to test for confidence intervals like you're saying. And I forgot the second part of your question, but we can obviously talk afterwards.
It's okay, thank you.
Let's move to, okay.